Hey guys, so I just kind of wanted to walk you through how we do the overall fractionation process. So when you get our filter drum, this will be inside of this so it doesn't break during transportation. So it'll be in there as well as this and the plugs. Everything will be inside. All you do is just screw this on. This is where the tag-on tubing will connect to the pump. Once you get that on, make sure these, I don't know what they're called, but just make sure those are on there properly. Keeps the seal nice and tight. Put this on top of that. Make sure it locks in with here. This is where you're going to put the filters. So we use the 25mm diameter filters. Just put one at a time on here. It holds up to 12. And then put the top on, make sure it aligns with that. And then you just screw it up. Make sure it's really tight, otherwise the water will seep through and contaminate all the other filters. So right now I'm going to go over how we begin the fractionation process once you get the samples. So this will be labeled, I'll send 3 of these with you guys, or actually you only really need 2. One will be the EPA composite, but you guys make that in the chemistry lab, and then you can just grab, I'll add a couple extra beakers for you guys that are clean and acid washed. So you'll have your sample. What you'll do is you will just pour it into the whole chlorophyll plastic beaker. You need about 500 mLs just to be safe. So this is your whole sample. We do need to pipette into here. So we pipette 15 mLs for each filter. How I normally do it is I'll have, because we do 2 duplicates for each one, for each station. 1 and 2 will be your whole chlorophyll A and B. Once we fractionate, this will be your less than 20 A and B, and this will be your less than 2 A and B. So for each depth, you will have 6 filters. Now getting to the actual fractionation process, it's actually pretty simple. Once you pipette your whole chlorophyll into the drum, you can then start fractionating. So you will have a 20 micron filter. All it is mesh, and it will drip, so be prepared for that. You don't need to attach this to the pump. As you can see, gravity kind of does it for you, which is kind of nice. You will have to attach the pump to the less than 2 micron. Alright, and you just flip this upside down, and then you will pour your 20 micron water into the 20 micron beaker. Then you'll pipette your replicates into your drum, so in number 3 and number 4, if there were filters in there. Then you want to pour some of it into your less than 2. I actually don't have filters in here, it's just DI water and there is no filter, but you will have to connect the pump to this to help with gravity. We do not go over 10. The centimeter HG or whatever, so we don't break any of the cells. I would, just as a tip, make sure that you have some left over. Don't pour the whole thing in there, just in case you do have to redo your fractionation. Like maybe the filters got contaminated somehow, and it's just kind of in your best interest and to save time. And then you just pour it in here. And then you have your fractionated water. And then you just fractionate into your filters. Say you're on the nearshore station, we only collect water from the EPA composite for nearshore. So you would need to use these plugs for the other 6 holes. That way the pressure isn't disturbed or anything like that. Alright, then after all the water is filtered out of the drum, you just take this off carefully. Sometimes the filters stick to the other side, you just have to kind of be careful. And then you would take tweezers, I use 2 tweezers, fold it once, and then you'll stick it into our little pop-top things. And then try and put it in the freezer as quick as you can downstairs, in that -20-degree freezer. That's really it as far as fractionating the chlorophyll. I'm going to also go over with you guys the labeling. I'm pre-labeling everything as much as I can, but I'll show that to you guys right now. Okay, so I'm going to kind of walk you guys through the layout of some of these cryotubes. I know it can be confusing, especially looking at other people's stuff. So everything's already pre-labeled besides the stuff that's highlighted. So we do need the date for when you collect the data, obviously. And then the depths for the Fmax depth and the B-2 depth. That'd be super helpful for us. But I really kind of just want to go over the way this is laid out. So all of them are pre-labeled. The one on the left, it goes from near shore, middle shore to offshore. So near shore, I have these marked for when there's a new depth. So the line just indicates, okay, these next 6 are going to be a new depth. And they're all pre-labeled for you. You don't have to write anything on these. So this is a racine transect sample that I'm kind of showing you guys. So it's marked as racine near shore, the epi depth. So whatever epi composite you guys do, if you could write the depths, that'd be awesome too. Or just let me know. And then WA. The W stands for whole chlorophyll. And then A stands for duplicate A. And then this is the same exact thing, it just says WB, so whole chlorophyll, duplicate B. And they just go in order like that. So this is whole A, whole B, less than 20A, less than 20B, less than 2A, less than 2B. So when you go to get your filters out, let's say I have 6 on here. This would be whole A, whole B, and it would go in those first 2. Then you'd have less than 20A, less than 20B, and it'd be the next row. Then less than 2A, less than 2B, less than 2A, less than 2B. I hope everything's pretty clear. It took me a while to definitely write everything out, but this would be the middle station. These will be the epi. So they're all nicely labeled for you. And then these 6 will be the Fmax, wherever you guys think that the highest chlorophyll concentration is in the deep chlorophyll layer. It's all nicely written out for you guys. And it's all in order, so you don't have to worry about it. And then this is the offshore for racine, epi, whole. This will be the Fmax tubes. And this will be the B- tubes. I mean, if you guys have any questions and you have cell phone service, feel free to give me a call. I'm going to send you guys a page, like a instructions sheet as well. But I figured having videos is a little more helpful. The next thing that would be super helpful if you guys did was to save whole samples for flow cytometry, and I'm going to show you guys that right now. Alright, so this box contains flow cytometry tubes that are also pre-labeled. I don't know if you guys have noticed, but everything is color-coded, so everything that comes from the Joaquin transect is green. And these are all labeled. These ones have to have some paraformaldehyde added, which is in the box that we sent you guys. And I'll have the volumes. You don't have to worry about calculating anything. I'll just tell you what volumes to add. But yeah, these need to go in the freezer right away. I would almost do this first when you get the water, just because it's easier. And these need to stay in the freezer, and when you send it, everything needs, all the samples need to have dry ice in them. That'd be really helpful. And then we could reimburse you guys too. So yeah, that's all I really got for you guys. Besides the bottles, I'll go over that right now. So the bottles are going to come in a cooler kind of similar to this one. It'll be a little bit bigger. But everything's color coded. So the smaller bottles will have glutaraldehyde in them. The only thing that needs to get labeled on these bottles is the date and the depth where you collected it from. So for example, I just pulled this one out. This is Joaquin Offshore, and this is the F Max, wherever you guys collect that water from as far as depth. All you have to do is write the date and the depth, add the sample, which is 250 mLs, and add the glutaraldehyde, which will be 5 mils of glutaraldehyde per glute bottle. They are all color coded, so Joaquin is green, Racine is pink, Saugatuck is orange, and St. Joseph is white. There's 2 sets of bottles per depth that need to be collected. We have 250 mLs, which I just showed you guys, and then we have 500 mLs, and this is for Lugol's. Again, everything's color coded. The only thing you have to add is the date and depth, the Lugol's and the sample. So hopefully we make this fairly simple for you guys. We super appreciate you guys collecting these for us. And like I said before, if you guys have any questions or anything, I'm gonna make sure I have my cell phone with me while we're on the island, as long as I get service. If not, you might be better off emailing me, and I might be able to respond sooner, because sometimes you get Wi-Fi, I guess. But yeah, yeah, thanks guys.